We have two speakers. The first is Rob Nichols from the LMB, who's been a long-term contributor to CSP4, but like all crystallographers is discovering the joys of cryo-EM, and he's going to talk about model building and refinement. Rob?
Thank you. I'm going to talk about fitting models into cryo-EM maps, or model refinements against cryo reconstructions, however you like the semantics. I'm going to talk about refinement, restraints and regularization, focusing on jelly body restraints, external restraints generated by ProSmart, those generated by libg, and also how you can use the external restraints in COOT for visualization and refinement. Then going to talk about a couple of other things that are relevant to CSP-EM, map sharpening and blurring, automatic box size, what that is will become clear, and a new divide and conquer pipeline, which will be released quite soon. I'm also going to talk about a couple of other COOT features, juggle fit and model morphing, which is very relevant to the topic. I should note that we are not talking about map interpretation, fold recognition, or model building here. This is purely about refinement. So we're making the assumptions that a preliminary model has been built already and has been placed sufficiently well, sufficiently close to where it needs to be in the map. The objective then is to refine the model, optimizing the fit between the model and the observations. So there's various software for this. We've heard about some today already. We've got Flexium, and Paul's going to talk about the offerings from Phoenix later. There are various software to do it, but I'm just going to talk about those that are being developed in the LMB, in our group. We've got RefMac, which is the powerhouse of macromolecular refinement. We've got COOT for visualization, model building, and fitting. We've also got ProSmart for the generation of external restraints, which are then used by RefMac or COOT in refinement, and also for comparative structural analysis. And also libg, which is for the generation of generic nucleic acid restraints. So what is the purpose of refinement? The purpose of refinement is to fit an atomic model into observed data. So the model, as we got here, should agree with the data, and so we're trying to fit it so that they do agree particularly well, and then refine the model so that it is chemically and structurally sensible whilst being consistent with the data that's been observed. Now, the structure factors are what we are treating as the observations. They're not actually the observations obviously are the projections, but from, say, RefMac's point of view, the observations are the structure factors, or the maps. It doesn't matter whether we're talking about reciprocal space or real space here, refinement in both is equivalent. There are quite a few differences between cryo-EM and macromolecular crystallography, obviously, but from a practical point of view, some of those really have to be considered. These software have been developed over a long period of time for use in macromolecular crystallography. And so if we're going to try to repurpose them for use in cryo-EM, we need to be very conscious of exactly what differences we need to account for. So some of the relevant differences are obviously that we are able to obtain phase information, which is fantastic. We're no longer dealing with a phase problem. Instead, it's a resolution problem, perhaps. This means that the map is not updated as the model is refined. That's one huge difference. The map is always static, if you like. That may not be the case in the future if we start doing co-refinement of the model along with reconstructions, but I think that is not on the doorstep just yet. Because we haven't got crystals, we're not dealing with crystallographic peculiarities, twinning, etc. We haven't got to worry about crystal contacts and what effect they may be having on the conformation that's observed. And also the boundary is not enforced. We haven't got a unit cell. We actually choose how, if you like, we choose how big the box is around our structure. And also the concept of resolution is really quite different. So in macromolecular crystallography, we talk about resolution, which is actually just the quoted diffraction limit, the limit to which the X-rays diffract. Whereas in cryo-EM, the concept of resolution is quite different, and it's much closer to the how it's used in the field of image processing. And further, we can actually consider local resolution. One similarity between them, though, is that due to them both being from scattering, we've got high resolution information loss. So the low resolution information is there, we can utilize that, but it's the high resolution details that is more of poor quality. And it's there where we really have to focus on injecting the right sort of information in order to stabilize the refinement. And given that most of the methods that we developed in crystallography are for that purpose, we can very readily transfer them over to cryo-EM. So RefMac 5 uses a maximum likelihood approach. The target functions have two components. We've got the, let's get that. There we go. We've got the likelihood of the data and the probability of the model. Now, the likelihood of the data is the negative log probability of the observations given the model. What that essentially represents is the consistency between the current state of the model and the data. And then we've also got the probability of the model, or the negative log probability of the model, which is all of the information about the model. So we've got the data to refine our model against, that's the structure factors corresponding to a given reconstruction, and we've also got the parameters to refine because we know that we're starting from a model that's already been created. We now need the prior knowledge. We need to construct the knowledge that can actually go into the system in order to help ensure chemical and structural integrity of the model at the end. We don't just want to fit into the density and completely ignore any of the chemistry. So what restraints are used to do this? We've got bond lengths, angles, chirals, planes, some torsion angles. We've got B-values and also van der Waals repulsions. And note that RefMac generally deals with restraints, not constraints, although there are some exceptions. I mean, you can if you want to. But generally speaking, we're doing restrained refinement. And so it can only really be used for high-resolution cryo-EM. So whilst we're able to push the low resolution limits with these tools in macromolecular crystallography, in cryo-EM, we're talking about the very high-resolution end of the scale, so maybe 3 to maybe 6, 7 angstroms is what we're talking about. So what do we know about macromolecules? So firstly, the macromolecules consist of atoms that are bonded to each other in a specific way. And that's, of course, specified by the current state of the model. We've got that the oscillation of atoms close to each other in 3D cannot be dramatically different, and so there we can introduce B-factor restraints, and also perhaps TLS restraints, although not so much anymore, moving away from TLS restraints. And if there are two copies in the same molecule present, then they will likely be similar to each other. And so for that, we can use NCS restraints. And they might be local or global, depending on the resolution that you're working to, and other factors. If there are two molecules with sufficiently high sequence identity, then it's likely that they will be structurally similar, in which case we could generate external restraints to take information from a high-resolution homologue and inject that in the structure that you're trying to refine. And also proteins tend to form secondary structures, and so you can also generate generic hydrogen bonds or secondary structure restraints for those cases. And given that DNA and RNA tend to form base pairs, stacked bases tend to be parallel, we can generate restraints for these using libg also. I'll just say something about NCS. There's three ways of dealing with NCS. One is that you can use NCS constraints, which simply says that the NCS related copies are considered to be exactly the same, and we want only one set of atomic parameters to be modeled, to be refined per molecule. And that's often what is done already in cryo-EM. We've got global NCS restraints, where the molecules are superimposed and differences between the corresponding atoms are minimized. But that's actually that's the way it was done a few years ago. But now we tend to always favor these local NCS restraints, where we assume that the molecules are only locally similar. However, they may adopt slightly different global conformations, and so we restrain the differences between local interatomic distances. And these interatomic distances would be typically up to about 4.2 angstroms. So that's very high resolution. So again, it comes down to injecting high resolution information and less focus on the low resolution. So why are we introducing so many restraints? So the answer is to improve the observation to parameter ratio, quite simply. Let's just take a very simple example, fitting a line where we know that we've got 2 parameters. Suppose we've got just 3 observations, and we're trying to fit a line. Okay, we can do that. Now suppose we've only got 2 observations. We can still fit a line, but the line's extremely unreliable. If you move one of those observations, then the line will change quite dramatically. And that's overfitting. That's going to cause model bias. Now, if you've only got 1 observation, well, you can fit a line anywhere. You can fit any sort of line that you want. There are insufficient observations, you've got unstable refinement, and it's an ill-posed problem. And unfortunately, that's the problem that we're dealing with all the time. We've got so many observations, and we've got way, way, way more parameters that we're trying to fit. So how do we deal with it? One way to deal with that is to reduce the number of parameters. So typically we're talking about 4 parameters per atom. We've got 3 coordinates and 1 B factor. Alternatively, we could do rigid body refinement, so we've just got 9 parameters per body, where a body might be a chain or a domain. But ideally, we don't want to do that. We want to do full model refinement, full atomic model refinement. So instead, we can increase the number and the strength of restraints. We can use B-value restraints, NCS restraints, H-bond secondary structure restraints, restraints to homologous known structures, nucleic acid and base stacking restraints, and jelly body restraints. This is all the use of prior knowledge to stabilize refinement, and this is what we call regularization. So what is regularization? Suppose we've got some sort of energy function. This is a function that we want to refine. We've got a starting position. Ideally, we want to get to the minimum. However, we might get stuck in some sort of local minimum somewhere and not be able to find the global minimum. Here's an artificial example where we've got a function. And suppose we're trying to, we've got a starting point over here, and we're trying to get to the minimum of this function. For that, we need the derivative at that point and the curvature at that point. And hopefully we can get to a new point to try, which should be closer to the minimum. The problem is that if we've got very noisy data, if we've got poor observation to parameter ratio, then those directions, those estimates can be quite unreliable, in which case we could be going in any sort of direction. But we don't want that. Instead, we introduce information, restraints, if you like. So if we know that the distance between x and y in this synthetic example is 4, then it changes the nature of the function, changes the nature of space, so that hopefully the search direction is much more reliable. So what sort of regularizers can we have? We've always got the geometry restraints. We can also introduce B-value restraints, which are actually default now. We can use local NCS restraints and external restraints wherever they are available. We can also use jelly body restraints, which are a special class of restraints. They're regularizers without a target value. I'll just go into what jelly body restraints are. So this is the function that is used for jelly body restraints. The whole idea behind them is that the model doesn't want to move too much in any given cycle. So what we say is that the distance between 2 spatially proximal atoms should not be too far from the current distance between those 2 atoms. Now this is actually updated every single cycle. So you've got the distance minus the current distance, which of course is 0. So actually these jelly body restraints don't actually add anything to the refinement target. Okay. Now if we take the first derivative with respect to that distance, and we again get distance minus distance, and so that's equal to 0. However, if we take the second derivative, we look at the curvature, that's actually non-zero. So what we're doing is we are introducing something which doesn't change the likelihood function or its derivative, but it does change the curvature of space. So it doesn't change the nature of the likelihood function, but hopefully instead of having a very noisy, unstable refinement, we should be able to stabilize it anyway, which sounds a bit like magic, and crazily it works in a very large number of cases. So here's the CCP-EM interface for RefMac. And if we look in the refinement options, there we've got jelly body. Let's just make that a bit bigger. And it's actually enabled by default in the interface. That goes to show how good we think that jelly body restraints are. They're not injecting anything external, they're just trying to stabilize refinement, and so why not use them? And that brings me to the use of ProSmart for external restraint generation. And the whole idea is the injection of prior knowledge to aid new structured determination. So given that the PDB has evolved and it's got so much of a wealth of information in it, why not utilize it? If you've got a structure that you're trying to create a model for, and there are known homologs in the database that perhaps are higher resolution, then we can utilize the local information from those structures in order to improve the local geometry in your structure. We can also, for the chains that you haven't got a homolog for, generate hydrogen bond restraints. And these restraints are utilized by both RefMac and COOTS. Importantly, because we're talking about local information, again, high resolution information, these restraints are independent of global conformation. So it doesn't matter if your structures are in completely different, if your homolog is in a completely different conformation to the structure that you're actually trying to refine. It should work as long as the local structure is actually the same. So here's a depiction of how it works. Suppose we have got an atom here in red in the structure to be refined, and we know that that corresponds to this atom here in a known similar structure that you've identified. And what we want is that the distance between two atoms in the structure that you're refining to be quite similar to that you've observed in your high-resolution homolog. So we can identify that and just inject it as a restraint. As simple as that. We go through the whole structure, identifying these interatomic distance restraints, or as much as you want. So suppose that we know that the distance between an oxygen and a nitrogen, so that there should be a hydrogen bond, so it should be roughly 2.8 angstroms. If we're starting off with 3.4 angstroms is the distance between them, during refinement that should change, and we end up with 2.8 angstroms between them, or thereas. That's the idea. So should you actually use it, is the question. If you've got a high-resolution homolog, aren't you just biasing your model towards that one? Well, yes, but let me take a motivational example from crystallography to hopefully convince you that it is worthwhile at least considering. So this is a structure from the PDB, so deposited at 3.5 angstroms. And you can see just by looking at the secondary structure that this is not a well-refined model. Not to criticise the authors, it was a while ago, so perhaps the software wasn't good enough then. But fortunately there is a high-resolution homolog, which is essentially identical in sequence. Slightly different global conformation. Certainly if you try to superpose them, you'll get that they don't superpose very well at all. But let's try to use this high-resolution homolog to generate restraints. If we refine the structure, just using default settings without anything, then the R factor goes down and the R free goes up, indicating awful overfitting, very poor refinement. So that's not good. Now if we use jelly body restraints, then what happens is that the R factor goes down slightly, the R free down slightly, but importantly, the difference between them stays pretty stable, indicating that we haven't got overfitting. Jelly body restraints aren't injecting anything from outside, anything external, but they are at least keeping refinement stable. The external restraints, however, to the high-resolution homolog, shake things up quite a bit. We can see some sort of conformational change which is happening where we've got bumps here, and ultimately you end up with improved R factors, both R and R free, whilst stabilising the difference between them. But you can't just look at R factors, we also want to look at the geometry. So here's the original Ramachandran plot from that structure, which is not fantastic. And here's the one after refinement using the external restraints. So just these restraints have been able to really tighten up the geometry. But we can't just look at refinement statistics and geometry. We should also look at the density in this case. So here's a particular region. Just look at the model and density as we refine the structure with the external restraints. So just show you that again. It's quite dramatic. the density for this region here has completely disappeared, and you've got a completely different interpretation of the density over here. So it's quite easy to just do a bit of real space with rebuilding in Qt. Then re-refine with the jelly body restraints, not putting the external restraints back in, just the jelly body restraints this time. We've got a completely different picture of this region. We can also have a look in the active sites where we've got focusing on, we've got this residue here, and also focus on this side chain here also. When we refine the structure with the external restraints, we get a completely different interpretation. So we've got this side chain changes position here, and we've got density for this side chain popping up here. It turns out that actually this is a tyrosine, and that lysine wants to be somewhere else. We now get rid of the external restraints and put the jelly body restraints back on, and refine the structure, completely different interpretation of the active site. So this is important. So when refining macromolecular crystallographic models at low resolution, we've got to check refinement statistics, but that's not always conclusive. We've got to check the geometry, that's not always conclusive, and the electron density, which is not always reliable. As we see, that can change quite dramatically. Fortunately, in cryo-EM, we haven't got that problem, because the map just stays the same. We take the map, comes from the 3D reconstruction, and we just refine against that. However, we could actually check the map's level of sharpness or blur. I'll come on to that in a minute. So the quality and suitability of prior information is also important. So it's always worth considering manual rebuilding of any high-resolution homologs before using them for restraint generation. Just check that there aren't any gross errors in there. And PDB redo is extremely useful for this. Maybe it'd be useful for there to be an EMDB redo at some point. So the conclusion from this is that external restraints from homologous structures can be useful, and I hope I've convinced you that that means that we can have confidence to use them in cryo-EM refinement. We also are interested in assessing the changes that occur during refinement. So what actually changed? So here's the original structure, and here's the structure re-refined using the external restraints. If we superpose the original model versus the re-refined model, we can see that although the global conformation hasn't changed at all, the details have. But how do we assess them? Well, we can use ProSmart for that also. So ProSmart's also for local structural comparative analysis, and also for the identification of rigidly conserved regions, useful for comparing both homologous structures before you try to refine them, and also models at different stages of the model building and refinement process. So the way it works is really quite simple. Just has a look at backbone main chain fragments, goes through the structures that you're comparing, identifying these. It superposes them together to identify the RMSD, and then colours the structure according to that local RMSD. And so you can see that all over the place there are red regions which correspond to large deviations in the backbone. We can also do for the same for the side chains. You see that the side chains, in this case at least, are just completely different to what they were before refinement. Now if we compare the re-refined model here on the left versus the high-resolution homolog on the right, we get a completely different picture. The global conformation is different. We've got domain motion there. We can't really see what exactly. However, when we colour the structures according to their local backbone conservation, you can see that the whole structures are white, apart from just a couple of surface loops, indicating that local structure is actually extremely similar between them. So the information from the high-resolution homolog has successfully been passed on. So I said that we couldn't really see exactly what the global conformational difference was, what the domain motion was. ProSmart actually allows you to investigate that as well. In this case, it identifies 5 that are coloured in here, regions which correspond to like the really rigid cores of conserved structure between these two structures. If we superpose based on the first one, we get that picture. Superposed based on the second one, we can actually see the way in which the loops are moving different between them. We can also see that they superpose extremely well within that individual domain. Similarly, for this one up here, we've got differences in the surface loop. Interestingly, in this domain, it's identified that there are two rigid regions that are actually in a different coordinate frame, indicating that there is some sort of conformational change here which actually involves that domain becoming distorted, perhaps to accommodate the rest. So that sort of analysis is useful for biological reasons. You might also be interested in, for example, assessing hinges. So here's one structure in the open conformation, one in the closed conformation. Colouring them according to this way, you can see exactly where the hinge is. And if you have a look at the identified rigid regions, we see there's the one region on the left and one region on the right, which interestingly does not include these two helices that you or I would probably have thought were part of that domain. Actually, there's a bit of a twist. They've actually changed conformation in order to accommodate that binding motion. So if you're interested in kind of the details, this can be quite useful. Anyway, let's move on. We've also got support for DNA, RNA, and you can obviously do the structural comparison in exactly the same way. So here we're comparing an EM model in blue versus an X-ray model in orange. You can run it in CSP-EM. Tom Burnley has very nicely created this ProSmart interface, which is very simple. You put your PDBs that you're trying to refine in there, and the reference homologs in there, and that's about it. There are other parameters that you can try, but essentially that's all you need. We can also visualize these restraints in Qt. So all of those lines that you can see there correspond to the restraints. Now, we can mainly see red there, but we've actually got three colours. We've got grey for very similar. So the low resolution structure is very similar to the high resolution structure in terms of the interatomic distance. We've got red implying that they're much longer in the low resolution structure, and blue implying that they're much shorter. And this is actually a cryo-EM map at 4.2 angstroms, I seem to remember. And you can also do full chain refinement now. So we're all used to refining just a couple of residues in Qt. Paul's actually working on essentially making it multi-threaded, so you can refine much larger regions in one go. At the minute, we're on 2 times speed up and using quite a few cores, and so it's got a long way to go from an efficiency point of view, but it can be quite useful. We can actually refine this whole chain using these external restraints, and it'll take a couple of minutes while you watch it going from cycle to cycle, but at the end, you'll get a picture a bit like that. So notice the colour change there. We've gone from red, so really different, to grey, where Qt has been able to, using these external restraints, pull the structure into the same conformation as in the homolog. But you don't always want to pull the whole structure into the conformation as the homolog. It could be that there are certain side chains or certain regions that are really different between the two. And so for that, we use robust estimation. So rather than using the least squares target, we use a plateauing function, which allows robustness to outliers. So using least squares, if you have an outlier versus a more extreme outlier, the relative difference that's applied to the target function is quite large. Whereas if you compare the same difference using this Jim-McClure function, it's much smaller. So that should allow robustness to outliers. It doesn't matter if you're an outlier or an extreme outlier, you're not going to make much difference to the target function, is the idea. So here's a case we've got a structure before refinement, and that is a homologous structure, a higher resolution homolog, and you can see that those side chains are in different conformations. And it should be in that conformation, it should not be in the conformation of the homolog. If we try to refine that structure, we get this, which is fantastic because the model has stayed in the density. And after refinement, we see all of these red lines indicating where refinement has not pulled the structure towards the conformation of the homolog. If we overlay them all, you can see them all there. We've got the original structure in blue, after refinement in yellow, and the homolog in green there. Importantly, though, it's actually tightened things up elsewhere. So if you look down here, we've got the original structure in blue, tracking there. whereas the main chain of the refined structure in yellow is much more similar to that of the homolog. So it's able to really tighten up the backbone in this case whilst leaving the side chain in a sensible place. Brilliant. We've got a very similar situation here. We've got this tyrosine, which is clearly in density, and the homolog, the tyrosine, is somewhere completely different. So if we try to refine them using the external restraints, we see that lots of grey where the backbone is, but lots of red around the side chain which has not been pulled during refinement. But what if there are no high-resolution homologs? We still need to stabilise refinement, so instead we can use either jelly body restraints that we've discussed already, or we can use generic external restraints, so either ProSmart hydrogen bonding restraints, or alternatively LibG for base pair or base stacking restraints. So this is the hydrogen bonding restraints. You can see you've got hydrogen bonds along the helices and between the sheets. You can also have them for the loops if you want, but it's all just for the main chain. And LibG, which is created by Fei Long in our group, can generate base pair restraints, so restraints between the base pairs to ensure that they are making sensible contacts, and also base stacking restraints. I'm not sure if you can see here, but there are lines between those bases saying that they're nice and parallel, which you can turn into nice big pictures with lines going all the way around them. Anyway, here's an example of a 3.2 angstrom model, where before refinement this base is clearly in the wrong position, and after refinement using these restraints the base becomes more parallel in yellow there, which is what we want. Which brings me on to something completely different. Let's forget about restraints now and focus on the map. So this is the default map of a 2.2 angstrom cryo-EM model. Yes, whether or not it is 2.2 angstrom is a question. But anyway, so this is the model that's built in, and we're just looking at the solvent pocket. You can see all of that noise in the solvent region there. If we blur the map by 20 angstroms, then a lot of that noise disappears. And you can still see the features in the map though. We blur by another 20 angstroms, we're on to 40. And we've got a much clearer picture as to what is going on, at least in my opinion. Go to 60, 80, 100 angstroms, and now we're starting to lose some of the finer details. We can now also blur back to 80, 60, 40, 20, the default map. And we can continue to sharpen if we want. We sharpen by 20, 40, 60, 80, 100, and now we've got absolutely no information about where the model is. There's very little difference between the solvent region and the model. Now we have the option, the ability to adjust this parameter, which essentially gives different weighting to the high resolution versus low resolution reflections, the structure factors. So how do we do this? How do we assess it? And how do we make a decision as to what is the most appropriate sharpening and blowing level to use? This is the CCP interface as of today. And we've got this thing here called MRC to MTZ, which has also been called RefMac SB and may change again. This is for, as it sounds like, converting MRC to MTZ files, but also allows you to sharpen and blur according to a number of sharpening and blurring levels. If you run this task, it will give you this graph, which is the average amplitude in a given resolution bin for each of the different levels of sharpening. So the default map is in green here. We see that it plateaus and kind of sits there at whatever that value is. But we know that the average amplitude should decay to 0 eventually. So perhaps we need to blur a bit. We've got green, orange, then red's the next one. So perhaps a blur of 40 is appropriate in this case. And true enough, if we have a look at the blur of 40 in this case, that seems to be quite a good map to be looking at. So that seems like a reasonably good strategy to follow. This is the same case, but zooming out, focus on this region here, where it's clear that we've got high B factors or something, or just not a good model. And if we focus in on it using the default map, we can see that they built something in, but it's there's really no evidence for it there. We haven't got a clue what should be there, even though in a nearby region we can have more confidence in the actual confirmation of the model. If we blur 20, blur 40, blur 60, we can actually see where that should be. We can continue to blur 80, blur 100, and so perhaps we're now blurring too much. But we can actually also display all of the maps at the same time, which is actually really quite effective. It means that you can see where the general, where the backbone wants to be going, as well as showing you something about the more high resolution features. And so that's always worth doing also, comparing with the default map, which is not always the best map. So blurring and sharpening is really useful for visual interpretation, and in macromolecular crystallography, map blurring and sharpening does not affect refinement. It's a post step. So you do your refinement, and then map calculation happens afterwards. It does not affect refinement at all. In cryo-EM, it does, or it can. I mean, you can generate a map afterwards, but you can also actually apply the blurring and sharpening before actually refining against that map. If we go to the RefMac interface in CCP-EM, in refinement options, and then somewhere down here, we've got map sharpen, which by default is not selected, but you can actually change that. So if we've decided that we need to blur by about 40, then we could actually put that in there and refine against it. But we've got to be careful that different regions of the structure may actually require different levels of blurring, which is one reason why it's a good idea to have a look at the array of maps. Because in some regions, the B factors may be higher than others, basically. So here's one particular case, and if we just have a look at this structure, coloured by B factor and Coots, and we can see there's a few regions here that are actually bright blue. Now, these correspond to extremely low B factors. So some of these atoms are around like 0.5 B factor, which is not physically sensible. And so you can actually refine this structure using a blur factor. So what this essentially says, if the B factors are that low, it means that the map's over sharpened, so it needs to be blurred. So we can blur it with 40 angstroms, colour it again, and we have something that might look a bit more reasonable. However, it is going to change the R factors. So if you re-refine the structure, we got blue before refinement and red after refinement, after 10 cycles of refinement, with varying blurring B factors. You can actually see that the R factor really changes quite a lot when you change this blurring factor. And that means that you cannot compare the R factors corresponding to different levels of blurring of maps. So that's one important thing to remember. If you look at exactly the same thing for the Fourier-Shell correlation, then you get a similar picture, but to nowhere near the same degree. So Fourier-Shell correlation is much more stable than R factors. In some circumstances, you might actually want to sharpen. So rather than blur, for example, in this case, we've got the blue model is the one that was deposited. Sorry, no, the green model is the one deposited. The blue model is actually just overlaid from a high-resolution homolog that was resolved later. In the original model, they couldn't actually build the model into that tube. However, if you take the sharpen map from RefMac, then you can actually see some sort of orientation, some sort of chirality to that tubey region, so you could perhaps see how that helix should be built in. So it's not always that blurring makes sense, sometimes both do. The other thing is that the box size, we've got a choice over that. In some circumstances, the box is actually much bigger than the model, in which case you can tell RefMac to automatically shrink down the box to around the same sort of size. We can do that in the RefMac interface by going into refinement options, and then down here we've got local refinement options and just a local refine button. If you do that, it will cut down the box size. Focusing on the Fourier-Shell correlation and R factors, before doing that, you can see that they are absolutely awful. After doing it, magically the Fourier-Shell correlation and R factors get much better. The other thing is the divide and conquer pipeline, which is to be released in CSPM imminently, perhaps hopefully the next few weeks. But the idea is that if you've got a really big thing that would take a very long time to refine, you can do it in parallel by essentially identifying an individual chain or region, identifying all the chains around that, and then just refining that one chain. And doing that in parallel for each of the chains in the structure, and then just building it back together afterwards. Really simple idea, but should speed things up. That'll be available very soon. So a couple of Qt features. We've already talked about what Qt can do with the external restraints in terms of visualization and actually refinement, and about how it can do the full chain refinement now using multi-threading. The other things that's worth talking about is jiggle fit and model morphing. And these for if the model is not sufficiently close for refinement straight away. Jiggle fit, the whole idea is to do a brute force search. The idea is to randomly sample rotations and translations of your model, transform the atoms, calculate density correlation, rigid body fitting the top 20 or however many solutions, and then select the best one. And what happens is that you've got your original model that might be in roughly the right place, and it should end up in pretty much the right place, but just using rigid body refinement. So we try all sorts of rotations, translations, hopefully get the right thing. But it is highly sensitive to translation. Whilst the rotation can be anything, the rotation of the starting molecule where relative to where it should be can be anything. Translation is quite sensitive, so you can only be a few angstroms before the successful jiggle fit really goes through the floor. But then once you've got your model roughly where it could be, you could then use model morphing, whereby for each residue in a given chain we say, where does the small molecule centered on a residue want to go? We identify all of those transformations and then apply them all on a per residue basis and repeat until convergence. So if we've got a region with a given residue, identify structure around it, identify the translation and rotation that that piece of structure wants to go to. And we do that for the whole chain, if you like. And then, in a robust way, apply iteratively. And so that means that if your starter model is not quite where it wants to be, then you can slowly go in, and that should be nice and flexible because the whole model's allowed to move. It's not rigid anymore. Okay, and that's about it. So, to summarize, restraint refinement can be used for cryo-EM. At least at high resolution cryo-EM. External restraints, the homologous structures, can be useful. They're used by RefMac and Qt. And if homologs aren't available, then you've got other options. Other things that you can think about are multiple levels of blurring and sharpening, which can help, but you've got to think about what you're doing. Care is needed. The box size needs to be selected appropriately, because that could be a problem if you've got really poor R-factors for shell correlation. The divide and conquer pipeline should be available soon, and use jiggle fit and model morphing if you're not at the stage of refinement quite. Looking to the future, I guess we could be utilising information about local resolution more, both in terms of dynamic map blurring, so that we haven't got to have multiple levels of blurring, and also restraint weighting according to the local resolution. If that information is available, why not utilise it? Parallel processing is a common theme. And also, looking to the future, we could have refinement against multiple reconstructions. And then much further in the future, we may even get co-refinement of model and reconstructions, but that's still a way off, I think. Similar to how PDB redo is fantastic, it might be good to think about EMDB redo at some point, so that we've got a resource of reliable structures, which can be then used in the same way that we are using high-resolution homologs from crystallography now to refine cryo-EM models. The other thing that I haven't mentioned at all is cross-validation. And thinking about the deposition of half maps is obviously, or other cross-validation methods, is something that we also need to do for the same reason, especially if we're thinking about EMDB redo. If you want any more information about this sort of stuff, then I would say the starting point is Alan Barron's paper from a couple of years ago, which outlines a lot of what I've talked about today, apart from the new things. And that's it. So, acknowledgements, well, to thank my group. We've got Gary It's RefMac, Paul Emsley, Coot, Oleg, who's been working on the CCP-EM interface, and also wrote LaRestra and the Divide and Conquer pipeline. We've got Fei Long with libg and Michael. From CCP-EM, thank Tom and Martin. Tom, in particular, worked a lot on the CCP-EM interface. Thanks to Marcus Fisher and Stuart McNicholas, who helped with the pro-smart structure analysis, and the CCP4 core team. Colleagues from MRC LNB and CCP4 are always really useful. We've got not only Alan, but also Jude and Anna for coming to us with problems, and meaning that we actually develop these tools to help them. And Jake and Toby for computing support. We've got a website with more information and these papers. And thank you for listening.
Okay, thanks very much, Rob. Time for a couple of questions.
I got two questions, actually. So one is, I'm surprised you use R factor now. Historically, people have said bad things about R factors. So should you be using R factors in this kind of...
Going back to when I was talking about... Yeah, it's here. So we've got... There we go. That's the graph that I'm looking at. We've got that the R factors are extremely unstable to changing the blurring. However, the Fourier shell correlations are not. They seem to be much more appropriate. I think that because R factors have been used in crystallography for so long, we're kind of stuck into that rut. People expect you to be talking about R factors. But certainly looking at Fourier shell correlations seems to be the way to go, certainly in cryo-EM.
So is it not good? Also, the Fourier shell, this is also an overall number. So shouldn't you be reporting alongside, like a local correlation as a function of just a...
Absolutely. I mean, this is averaged over the structure, which, again, we're talking about global statistics. We shouldn't only be looking... I mean, global statistics are useful, but we should be looking at local structure also. You're absolutely right, yes.
So I hope we get away from the R factor. But so the other question is, you discussed the in the blurring and the sharpening, and the fact that you see actually at lower if you blur more at lower resolution, you see features coming up, which is very common to at low resolution we see glycans completely, and you don't see them at high resolution. Mm-hmm. So in crystallography, I always thought that is actually related to our bad handling of the very low resolution refractions. So you miss the big waves, and that's why we don't see like the glycans anymore at high resolution. Mm-hmm. Now with EM, we don't have that problem. We do
Absolutely.
See. Yeah. So why do you still have that problem so much with this? And not because you do treat the low resolution. You have the low resolution data. So I don't see why you should have that problem so much.
Your question is why in cryo-EM maps do we still have problems interpreting the low resolution features? Is that your question?
Yes, so you showed this case where you had, like, if you blurred more, you actually see the features coming up.
Yes.
Yeah, which is very, very reminiscent of what you have, like low resolution glycans, for example. Then you actually see this at low resolution crystal structure. You see full glycans. You take the same structure, you have a high resolution data set. You don't see at the high resolution maps. You don't see anything of it anymore.
Absolutely, and you can see the same thing in for ligands as well, if you've got ligands which are reasonably flexible. I mean, there's a variety of sources. I mean, there's also issues with the reconstructions. I mean, I'm confident with classes as well. I think there's a lot of sources of errors, so actually track them down can be extremely difficult. In this case, I would say that it's simply a case of mismodeling. In this case, you've just perhaps got some flexibility there, and so you can see the low resolution. It's the high resolution features which are just not coming out in the map. But
yeah,
no, I'm not sure what else you could say.
Yeah, so it should be better. Yeah, we should have a better analysis of the low and high resolution features of these things. Yeah,
absolutely, and I think that comes down to utilising information about local resolution also. I mean, if we had that information, then that could be used in deciding about what sort of maps we should be looking at, and how reliable the different high versus low resolution information is in a given area. Yeah. And I think that you can't do that with crystallography. And to be honest, if you could, it probably would have been done already. I think that cryo-EM is a fantastic tool for not having a lot of the problems that crystallography did or does.
Can you just say what those numbers mean, the blurring numbers?
The blurring numbers.
What do they mean?
A B factor. So if you imagine you
Just
add
B factors.
Yeah, absolute B factor. So we're simply adding a B factor to the positions.
And can you do that on a local basis?
There's no reason why you can't do it on a local basis. It's not done today, but there's no reason why you can't. That's what I was saying. If we had information about the local resolution or some sort of uncertainty, whatever you want to call it, some sort of measurement of uncertainty or reliability of a given region, then you could blur it differently. At the minute, the blurring is done in a reciprocal space, which means it's applied to the whole map. It's just convolution.
So, any more questions?
I think we.
So kind of coming off that local resolution question, with Relion 2, you can kind of locally filter your map to the local resolution. Mm-hmm. Would you be able to use those kind of locally filtered maps in the RefMap refinement and kind of handle a kind of multi-resolution refinement?
Multi-resolution refinement. So are you talking about the blurred maps? The different levels?
So kind of could you use like a locally filtered map that you're getting out of, say, Relion 2 instead of kind of applying a kind of overall blurring factor?
RefMap will use whatever map you pass it. And
okay.
So if you've processed the map, then RefMap will fit into that map in exactly the same that Qt will.
Okay.
So that's one big difference actually between RefMap and Qt, MX versus cryo-EM. That both RefMap and Qt are now just fitting into the map.
Okay. Yeah.
Yeah.
Thank you.
Okay, so I think we should thank Rob again and move on.